Hey again, it's me, Stephanie, and we are here to talk about gymnosperms, continuing on our walk down the kind of evolutionary story of plants. So we started algae, then we talked about those first land plants, then we talked about the first vascular plants, and now we're going to be talking about the first seed plants. And these are the gymnosperms. So here are today's learning goals. First, to be able to recognize and know the function of the different components of a gymnosperm sexual cycle as demonstrated by conifers. Then be able to recognize and know the function of different vegetative characters of gymnosperms. Then be able to relate the reproductive and vegetative characters of gymnosperms to their evolutionary histories. And then be able to recognize conifers, cycads, ginkgo, and gnetophytes as gymnosperms. Just realize these learning goals are definitely not in order of what we will be talking about. In fact, they're sort of in reverse order. And here's your vocabulary list. Now, these first few, we've got Pinophyta, Cycadophyta, Ginkgophyta, Gnetophyta, that G in Gnetophyta is silent. Those are actually all, we're going to call them phyla. But they're known as sort of divisions, which is just a more inclusive taxonomic rank. And a lot of these other terms, well, some of them will look familiar to you, some will be new. I need to tell you that that term seed, this is a term that we are going to expand on greatly in the next video. I'm going to kind of black box it this time, where we're going to I'm going to tell you about its importance, but we're not going to talk about its anatomy until the next video. So here are some gymnosperms for you. Now, gymnosperms really evolutionarily took over the planet sort of after the Lycopodiophytes had dominated the planet for many, many millions of years. The gymnosperms had this very, very special sort of way of dealing with their babies, which was by putting them inside of seeds. And this was extremely beneficial for them. And it increased the reproductive success of gymnosperms by quite a bit. In fact, so much so that they really outcompeted the Lycopodiophytes. And most of those, virtually all of the old all of the really big Lycopodiophytes are extinct. Only those smaller ones remain, but there are plenty of very large gymnosperms, including some of these conifers we see in these pictures. So something right off the bat we can see is they can definitely deal with pretty intense environments, including very cold temperatures for very long periods of time. Notice these trees, the picture on the left here, these trees that are covered in snow, they actually still have their leaves on them. So they've actually figured out a way to deal with maintaining their leaves in these extremely cold temperatures. And then this is just a cool picture showing what those trees sort of look like in the summertime when all that snow has melted. If these woody trees are growing on a slant like this, the snow sort of builds up, and it results in all of these trees sort of having a little arched base where the snow was so heavy and developing, just sort of pressing on the developing sapling of those young conifers as they were growing up. So they do grow up. They don't start big and woody. The reason why they are so successful relative to the Lycopodiophytes is because they have sort of seeds instead of just these naked spores. And then, in the Lycopodiophytes, those spores would develop into these Gametophytes that were dependent on a very specific type of environment, right? The Gametophytes were usually, like we saw in like the ferns, for example, they're these sort of little haploid structures that actually they needed to be in wet environments so that the sperm cells could swim from one gametophyte to another, essentially, around in the environment. And we'll talk about how these seeds deal with that. Anyway, a seed of a gymnosperm will germinate into a sporophyte, and maybe eventually, let's see if this works, it'll grow into something absolutely massive like this. So here's just a really big redwood tree that I saw this summer in the Prairie Creek National Park. We are so lucky to have these relatively close to us here in Humboldt State University because redwoods are known for being the tallest trees in the world. They can be quite massive. And one of the other reasons why they're so successful besides seeds is that they have grown true wood. They have evolved true wood. Here are some examples of gymnosperms that are extant, that are still living, that we're going to talk about very, very briefly. These are things that you can see in the HSU greenhouse. And so after, COVID regulations are eased up eventually in the future, you all have to go to the greenhouse. You have to go there at some point. You must. And see all of these things that I'm about to talk about. Now, this picture on the left, the Araucaria, the Araucariaceae, that's the family, you don't need to know that word. But this word is one of those words from your terms list. Pinophyta, this is that phylum, and it's really the phylum of conifers. And this is just a picture of a conifer here on the left. We've got cycads, these sort of look like palm trees. Ginkgos, these make really stinky seeds. And Welwitschia, this very strange looking plant. I'm going to talk about those really briefly. So with that really brief intro of diversity, like I said, we're going to talk about those here in a sec. But this is a cladogram that we have this is a new cladogram, but we're sort of building on the cladograms that I've shown you in previous videos. Notice that you could really kind of connect them into one larger cladogram in your notes or something like that. And I highly recommend sort of having your other cladograms nearby as we're talking about these very, very briefly. First of all, there's something that's sort of different here that we haven't addressed about cladograms. Sometimes these nice binary splits, as you can see, most of these common ancestors here just have sort of two lineages, one to the left and one to the right. But then we have this common ancestor right here that has exactly 3 descendants coming off of it, appearing and apparently at the exact same time, sort of. It looks like they all evolved at the same time, according to this cladogram. But the truth is that folks just don't know exactly which came first. Now, one of the concepts of evolution is that really all speciation events were probably more or less binary, where we had a population that sort of splits into 2 populations. That's the running theory for most speciation events. But in this case, where we've got these 3 descendants, basically we just need to do more genetic studies to get that unresolved node resolved, so we can know which of these 2 are more closely related to each other. Now, in fact, there have been some recent genetic studies showing actually that conifers are more closely related to Gnetophytes. This is what we're going to go off of right now, but just know that if you look in the literature, there are other theories other hypotheses posed about the relationships between these organisms. And that makes sense, because remember, cladograms are hypotheses. We're doing our best to figure out what's the truth, and one way to figure out to kind of get closer to the truth is to, let's map some traits onto these and see if they make sense. So let's see, we already talked about how the seedless vascular plants, so the things like the horsetails and the ferns and the club mosses and the spike mosses, remember Selaginella, Lycopodium, those were all part of the seedless vascular plants. And they, like it says there, they sort of evolved xylem and phloem, vascular tissue. Cool. And so that means that from this point sort of onwards, all of the descendants of the common ancestor right here had vascular tissue, had xylem and phloem. Cool. But the gymnosperms evolved sort of fancy. They did something fancier with that xylem. They were the first to evolve a vascular cambium. So in other words, secondary xylem. So the gymnosperms, these are our first plants with true wood. Remember, true wood literally just means secondary xylem formed on the inside of a vascular cambium. So along with this, true bark also came a little later. So let's write in vascular cambium. That implies secondary xylem. Now there is some evidence that actually the Gnetophytes, these are a group we're about to talk about, but they're in the gymnosperm group, that these Gnetophytes are more closely related to the angiosperms than the other gymnosperms, namely than the ginkgo, the cycads, and the conifers. The evidence for this is that both the Gnetophytes and the angiosperms have true xylem vessels. Remember when we learned about xylem and phloem, the sieve tube elements were part of the phloem. And then in the xylem, we had tracheads and we had vessel elements. Well, there are actually different types of cells too in xylem, but true vessels, those really girthy cells that allowed an incredible amount of water to be passed through them, those most likely evolved sort of before the common ancestor of the Gnetophytes and the angiosperms, so right here. True xylem vessels. So before these true xylem vessels, there were just other types of xylem cells, tracheads, other things. Okay, so that's about the vascular tissue, at least. What else can we talk about, though? I can change my color here. Some other things that the gymnosperms have that seedless vascular plants and bryophytes and algae do not have are, well, talked about this already, seeds, that's a big one. And with seeds, actually I guess kind of before that, they had the condition of heterospory. Remember the difference between homospory and heterospory? It's whether or not the sporangium produces one type of spore, and therefore one type of gametophyte, that's homospory, or if the sporangium produces two types of, or if there are two types of sporangia that produce two types of spores, and therefore two types of gametophytes. Well, yeah, the gymnosperms and the angiosperms have heterospory. Well, the thing about heterospory is that we know at least one member of the Lycopodiophytes has heterospory as well. But most members of the seedless vascular plants had homospory as their sort of sexual condition. So this one lineage within the seedless vascular plants did evolve heterospory, but that probably wasn't a close relative of the gymnosperms. And so in other words, that condition of heterospory, it must have evolved independently twice. So again, this would be sort of, if you have your other cladogram handy, you remember that heterospory evolved just on the way to one genus that we talked about. So imagine that this lineage that I'm labeling right here is the one for the genus Selaginella. Well, Selaginella was the one that also had heterospory. And so other members of the seedless vascular plants had homospory. And that's why heterospory is a convergence here. This is a prime example of convergent evolution. Yeah, where the same exact trait evolved multiple times. Maybe that's because it was beneficial. Probably. All right, so now I'm just going to briefly walk us through some examples of diversity within the gymnosperm group. And we're actually going to start with these ones, the ones that are most closely related to the angiosperms. At least that is the running hypothesis right now. Oops. Phylum Gnetophyta, or Gnetophyta, I hear it pronounced both ways. And again, really one of the main reasons why I want to share this information with you is just to show you some cool pictures of diversity, especially ones that we can see right here in the greenhouse at HSU, if we have access to it. So Needham is pretty neato. It has opposite leaves, so we know what that means. It's got 2 leaves per node. It also has non-flagellate sperm. So this is important only because the angiosperms are the other seed plants that do not have flagellate sperm. So the other groups of seed plants have swimming sperm, or in other words, sperm with flagella. Just like the evolutionary ancestors of the seed plants, right? The seedless vascular plants, the bryophytes, they all sort of needed to have swimming sperm because the sperm would swim from one gametophyte over to another one. And so swimming sperm might sort of be the ancestral condition here. Non-flagellate, non-swimming sperm would be the more derived condition that's shared with all of the gnetophytes and angiosperms. So here's another gnetophyte, Welwitschia. This is the definitely the coolest of the three that I'm going to briefly talk about here. Also visible in our greenhouse. This is a really cool plant. It only produces 2 leaves. That's it. In its entire life, it will only produce 2 leaves. They are exactly opposite from one another, as you can sort of see in that lower picture. They've only got 2 leaves, and if they're both kind of coming from one point, we can assume that's from one node. And sure enough, what happens is they have a basal meristem. So all of the cell division is taking place right at the base of those leaves. And so the ends of these leaves, these are the oldest parts over here. Sorry, you kind of can't really see it with my black. The very tips of these leaves. And that's just where they're old and dying. It'd be right in the middle that a reproductive structure would emerge if it was going to. And something that's noteworthy is that they're either going to be producing these male cones, like you see up in this picture, or they're going to produce female cones. So plants, the gnetophytes, they don't produce cones that have both male and female parts on them. And same with the entire plant. Like one plant of a gnetophyte will only produce male cones for its entire life, or it will only produce female cones for its entire life. Okay, moving on. The third genus of phylum Gnetophyta is Ephedra that I wanted you to know. These 2 have non-flagellate sperm, and they too have opposite leaves, just like the other members of Gnetophyta that we just talked about. But this time, the opposite leaves are not very noticeable. They're these little dinky brown things, like that you see kind of right here. And that is a pair of leaves, actually, at that node right there. So if we look kind of closely at the way that these stems are arranged, we've got a node right there. Oh, here, let me do it on a different one that's showing multiple nodes. So like this one in the middle, we've got a node right here and a node right here. And at each node, we've got those 2 brown leaves. The leaves are not doing any photosynthesis. Notice that. They're brown. They're crispy. You can feel them. They're papery. And they're really just vestigial, meaning they're more or less useless. Because notice their stems are green. They've got these elongate internodes that are responsible for photosynthesis. Cool. These also produce either male or female cones, just like the other gnetophytes. Okay, moving on to the next phylum that I wanted to talk about. This one's a really interesting phylum. And hopefully this helps kind of put some of the taxonomic rank terms that I've been talking about into perspective. This one phylum, Ginkgophyta, or Ginkgophyta is how I hear it pronounced sometimes too. It's a phylum in sort of in equal rank as Pinophyta or Gnetophyta, those ones that we were just talking about. And by equal rank, I mean sort of equal in terms of their common ancestry. So, do we have, it's not like they all evolved at the same time or for the same amount of time. It's just who are the descendants of some very old common ancestors. And it turns out that this is the sole living member, descendant of a very old common ancestor. This is the only species of this entire phylum. So like compared to Pinophyta, that phylum has thousands of species. And this one has just one. So not all phyla are equal in terms of their species richness, their numbers of species. It's a very, very, very tiny one. And it sort of has a cool story. It might have persisted throughout the millennia because folks used to plant them outside of monasteries. And so for millennia, humans have been propagating these sort of as ornamentals. Notice they're very pretty. They do change colors from green to yellow, orange, red, all the goods. And when you have a plant like that changes, that drops its leaves every year, sort of seasonally, it'll drop its leaves like usually in the fall, right before winter hits. That type of plant would be called a deciduous plant. So ginkgo is deciduous, and they have these really distinctive fan-shaped leaves. You see those? A lot of times they have this little lobe sort of in the middle, hence that specific epithet biloba. But other times they don't. So like this one, it's just more of a regular fan shape. Cycads. This is another phylum. Notice I don't have any genus or species names. Those were really were cool examples. But this one, there are plenty of genera and species of cycads, but I'm just sort of showing you a bunch of them here. And they too have flagellate sperm, so they have that ancestral condition of flagellate sperm. They have these really, really rigid leaves, and part of the reason for that, folks think, is that they evolved to sort of deter herbivory from dinosaurs. And so these leaves, you got to watch your eyes when you're walking around in the greenhouse near the cycads, because they are seriously sharp and very rigid. And I have definitely gotten scratched by one when I wasn't paying attention, like on my arm, and it bled a little bit. It's pretty intense. You don't want to get poked by these things. In this picture, we've got a bunch of these little tiny structures on these cone scales. So we've really zoomed in on a tiny cone scale in this picture, like sort of one of these things. And there's a bunch of these little structures. Well, these are little microsporangia producing pollen grains. More on that in a little bit. The cycads, they do tend to have this really palm look to them. Like a lot of times they are called palms. Now the truth is that palm trees are members of the angiosperms, which we'll talk about next week. These are very superficially similar to palm trees. But one of the big differences is these leaves are always going to be really rigid. Now palm tree leaves can also be pretty rigid, but they have a different sort of development. You can ask me that in office hours if you want to talk more about that. And then here it is. Another phylum. This is going to be the one we're going to talk about sort of for the rest of this video. This is the phylum Pinophyta, the conifers. And again, genus Pinus is what we're going to use as our example for a few things here. Members of this phylum, Pinophyta, we call them the conifers. A lot of times people call them Christmas trees or something like that. Or evergreens, which not all conifers are evergreen. But they all have non-flagellate sperm, and at least members of genus Pinus have needle-like leaves. But the truth is that we have a lot of variation of leaf morphology in this larger group, the Pinophyta. Sometimes leaves are not needle-like in this larger group, but they always are in genus Pinus. That's why I have it on this slide. So the other shapes you might expect to see would be very small green overlapping leaves or something like that. I'll show you examples of those at some point this week. So a pine leaf is pretty much what you would expect now that we can, there we go. Now that we can go back and think about the things that we learned those first few weeks in school, the stems and leaves and roots, right? All of these taxa that we'll be talking about from here on out have them. And so those terms like stoma, vascular cylinder, mesophyll, these are all terms that we learned in the past, and now they definitely do apply to all of the taxa we're talking about. Most of those terms we've talked about. So remember a stoma, you can see there it says sunken stoma. Well, yeah, the stoma are kind of deeply embedded here, and maybe that's a way for them to sort of maintain or sort of to reduce transpiration, like we've talked about in the past. Again, they tend to have a really thick cuticle, waxy layer. And what else do we have here? Oh, resin ducts. We've got these really large structures that are not part of the vascular cambium, right? Not part of the vascular cambium, not part of the vascular cylinder or the vascular bundle here. The resin ducts are these tube-like structures in the stems and in the leaves, probably roots too, just throughout lots of gymnosperms. And these are going to be sort of carrying antiseptic compounds, compounds that will reduce the likelihood of a fungus or some other pest clinging on to this organism. So it sort of helps them with antimicrobial issues inside their bodies. Okay. Secondary growth, we talked about this in great detail. And now we can just sort of remember those details. So, I know that your final exam, it's only over information from weeks 9 through 14, but that's why I'm really briefly reviewing some of the older stuff from this semester so that it can be allowed to go on the exam. So remember the vascular cambium, which forms in between the xylem and the phloem, right? So this is that ring, your vascular cambium right there. And then there it is in an older stem. And then there it is again in an even older stem. The vascular cambium is a very important sort of ring of cells. And sorry, to back up, this diagram is looking at sort of little cross sections through an aging stem. So this one at the bottom, this is the oldest part of the stem. And that vascular cambium has produced a lot of the secondary xylem, which is sort of pink or red in this picture, in these pictures. The secondary xylem is wood, and the secondary phloem is on the outside of that vascular cambium. Now, a lot of gymnosperms, they also have the whole the whole periderm thing going on too, which was formed from cortex cells. Remember, the periderms are the layers of cork cambium and its derivatives, and its products. So the cork and the phelloderm form the periderms, which is outer bark. Cool. And then remember, the nice thing about this is that nice from our human perspective, is that they end up with these yearly growth rings that you can see under a microscope. Actually, you can even see them with your naked eye, usually. And then you can count how old a tree is based on years. So you can see them in this picture too. You can really attribute one of these rings to a single year. And remember, this really darker wood in the middle is called the heartwood, and it's actually it's not transporting water and nutrients up and down anymore. It's only this sapwood that's actively conducting water. Okay. Remember, inner bark, this was our secondary phloem, and the outer bark, these are our periderm layers. I'm saying periderms because really each one of these is a layer of a periderm, which has cork on the outside and phelloderm on the inside. Good memories, I know. Okay, moving on. Here we go. Now, we have evidence of the evolutionary history of some of these organisms. Now, not a ton. Plants don't fossilize that well. And if you're interested in this type of stuff, Mihai Temescu has a couple of classes, actually. He's a paleobotanist here at HSU, and he studies, he has a class called paleobotany, and also the evolutionary and morphological history of plants. I might have butchered the name of that class, but in both of these classes, he talks about the evolutionary history and sort of the evidence that we have for all of these assertions that I've been making to you basically all semester long about evolutionary relationships. Well, a pro-gymnosperm is sort of an extinct lineage, or a few lineages, of plants that are intermediate in time in between the seed-free vascular plants and the gymnosperms. And so they sort of have intermediate strobili. Remember, a strobolus was that reproductive structure, the cone, and we can still use that word strobolus in the gymnosperms. So this is just maybe, and this is a scientific illustration rendition of this fossil. Pretty cool. Just some more fossil evidence. Remember those tetrads? Those conveniently do fossilize well. And so we have evidence that that's all real. Okay, so here's a heavy slide. We're going to talk about this in great detail. This is the first time we'll be seeing it. So the first thing I want to say is we have a lot of similar things between a lot of similar features between the life cycle of the conifers and the life cycle of some previous taxa we've talked about, like the Lycopodiophytes, right? We have some really, really familiar terms like, ooh, there's an embryo right there. That's a good one. We know what an embryo is. It's the result of, fertilization that, forms a zygote. So we've got all these same terms, okay? The embryo will go on to form the sporophyte. So it'll start off as a little seedling, and then it'll become a little sapling, and then the mature adult sporophyte will produce two things. It's going to produce male cones, and then it's going to produce female cones. Cool. Now, sometimes the male and the female cones are on different individual like different trees or something. But in a lot of cases, like in the pines, the male and the female cones are on the same individual. Okay. It's just that we've got male cones and these produce pollen, and then over here we've got female cones and these produce seeds. So, like I said, we're going to talk about a lot of these fine details here in a sec. I just wanted to just remind you that this is our good old-fashioned diplohaplontic life cycle, and again, it's we're seeing heterospory because we have two different types of sporangia. We've got the sporangia that will produce megaspores, and then we have the sporangia that will produce microspores. Okay. So we need to talk about what a seed is. I think that word seed might be the most sort of poorly understood word that's thrown around in everyday lives, as though people kind of understand its importance, its evolutionary history, but really most people do not understand that a seed is an amazingly incredible evolutionary benefit to the organisms that have it. What's that? And it's something that we need to talk about in an entire separate video. So we're going to talk about what seeds are later. Right now, I just want you to know that there are two types of cones, male cones and female cones. We call the male cones a microstrobolus and the female cones a megastrobolus. This jives with what we talked about last week, that microspores produce sperm cells and megaspores produce egg cells. Well, that is the case here. And so we just throw that word strobolus at the end of the prefix, and you've got yourself a male cone or a female cone. Notice that the male cones have all of this powdery, fluffy, cloudy stuff. These are actually bajillions of tiny, tiny pollen grains. And so like we said earlier, the sperm cells, they no longer need to sort of swim on the forest floor. They're now flying through the air, embedded inside of little pollen grains. It's very, very convenient from a conifer's perspective. Now, something you can't really tell here is the scale. A male cone is much smaller than a female cone. This is almost always the case. And sometimes the female cones can be like 100 times bigger than the male cones. And on that note, a lot of times there are way more male cones than there are female cones. There are generally fewer female cones. Here are what some male cones look like. At different stages of development. These are two different types of conifers here. Like I said, they're usually very small and lots of them. And here are what some female cones look like. Sometimes they are also very small, and then other times they are quite large. Sometimes, like in this picture right here, the cones will stay attached to the tree. So you see all these like kind of knobby things on the edges of these stems. So like here's a stem, and then there's all these, they're very small, kind of hard to see in this picture, but these little knobby things, well, these are cones that stay attached to the branches of these trees. And actually this happens to be a picture of a species that is dependent on fire. So these have serotinous cones, and the cones won't open. They'll stay attached to the tree like that, unopened, until a fire comes. And this is a way for them to sort of spread only in conditions that are convenient for them, or that are not convenient, that are favorable for them, which is fire. Yeah, some trees love it. And then this picture on the right, I just had to show you. This is what a bunch of cones that have been torn apart by squirrels look like. So this is like a few dozen cones. See, like there's one right there. We've got a few cones right here. And all of those cone scales have been removed by squirrels so that they could get the seeds. Yeah, squirrels love conifer seeds, a lot of them. And we've got all these sort of little pieces of tissue, these little brown pieces of tissue everywhere. Those are the cone scales. Those are what make up a female cone. And so that's what all of these sort of repeating units are that we see in this picture here. A female cone is, notice it's just sort of like the same thing. It's repeated over and over again. And this would be a side view of it. And here we have sort of an inside view of it. And this would be like a cross section through, right through the middle of one of these cones. And now we're looking inside of it. And we've got sort of that central part. This is the cone axis, where all of these thingies are attached. Well, all of these thingies, these are cone scales. These cone scales are what seeds are attached to. Didn't mean for that to end up looking like a flower. Okay, some more looks of cone scales. I mean, of female cones and their cone scales. Okay. Like I said, we're really going to talk about what a seed is in the next video. And so I just wanted to show you that at least this part of the life cycle, it's really important here. We have meiosis occurring in two different places on pine trees. One place that it occurs is in the microsporangia. These are also known as pollen sacs. And so all of these little tiny structures in there, those are the cells that are going to undergo meiosis. The cells that are undergoing meiosis are called, well, one of two things. They're either called a microspore mother cell, or notice if it's the female, then it's a megaspore mother cell. Or the other word for one of those cells that's going to undergo meiosis is a microsporocyte, or a megasporocyte. So those two sets of terms really are synonymous with one another. Either way, they're going to produce four haploid cells. At least right off the bat. So this is the nitty-gritty stuff that we're going to talk about in the next video. But just know that this tetrad of microspores, one of those microspores will become a pollen grain. Okay, so another word for a pollen grain is a microspore. Cool. And then there's just something else that I want to tell you now. Yes, there are 4 haploid cells that are the result of meiosis, those 4 megaspores, but only one of them survives. The other 3 abort, and we just end up with this one functional megaspore. And that's the one, I guess, down in this picture. This is the stage right before meiosis because we have the megasporocyte, or in other words, the megaspore mother cell. This is the cell that is going to undergo meiosis and then results in these 4 megaspores, of which only one will survive. Okay, so this is right here. This is an ovule. This is what we're going to talk about in great detail in the next video. So don't worry about the details of what's going on there right now. I just wanted to show you that inside of one of these female cones, like I said, we have these multiple overlapping cone scales, or in other words, an ovuliferous scale. Ovuliferous because they are bearing ovules, which again, this would be an ovule here. An ovule is a young seed. Okay, And so this is sort of one unit of a cone right here. And we have this unit repeating over and over again. And so we have this ovuliferous scale, that is this part. And then we would have an ovule. Ovuliferous scale, and an ovule. So subtending each sort of set of ovules would be an ovuliferous scale. And then subtending that, there's this other sort of structure called a bract. A bract is just a modified leaf associated with a reproductive structure. So in order to get those sperm cells to an egg cell, the egg cell is at the bottom of one of these cone scales, which is inside, deep inside of one of those cones. And so a pollen grain is going to have to really find its way to one of these. And first of all, it's going to have to, like I said, fly through the air. And, the lucky 1 in a billion or 1 in a trillion pollen grain will sort of fly through the air and land right here. And there's barely an opening. You can barely even see it. You can't see it with your naked eye, but a pollen grain is sometimes, the lucky one, like I said, is able to get in there. Oops, go back. And that's where it needs to go. Because again, our sperm cells are in the pollen grain and our egg cells are in the ovule. That's, of course, what we need. We know those two things need to meet in order to have fertilization take place. Okay, so we're going to talk about what the difference is between a young ovule and a mature ovule is. But right now, just know that one functional megaspore is going to go through mitosis, okay? Just like it did in Selaginella. The megaspore underwent mitosis and it became a megagametophyte. Well, it's the same thing here, actually. That megaspore will go through mitosis and it will become a megagametophyte. And actually, just like in Selaginella, that megagametophyte will produce archegonia. It does here too. And just as always, like we've seen since the bryophytes, an archegonia is going to produce eggs. Great. Okay, so anyway, pollination, those pollen grains flying through the air, that's occurring when this is the stage of development, before meiosis has even started to take place, actually, in the megasporangium. Crazy. Yeah. This is the stage. Meiosis has not even started to occur on the female side when it's sort of accepting pollen grains. And there it is, flying around, and it lands there, that lucky one. And then a pollen drop of sort of this sappy resinous stuff will sort of emerge from this little opening right here. And then the pollen grain will sort of get absorbed into that pollen drop and it'll get sort of sucked in. And then that's what pollination is. So when a pollen grain sort of enters an ovule, it more or less induces egg production. So that's kind of interesting that pollination happens far before fertilization will take place. In fact, it might be a year or so until fertilization takes place after pollination. Crazy. Yeah, and once that pollen grain is inside, it sort of closes up, and this is great. This is great. Oh, that's the end of my slide, so I will see you at the next video.